Hey, my name is Maester Teacher. How are you all doing? Last time I came, Daddy Gross did a marvelous job for us. Today, another colleague would be joining us.
My name is Miss Guyton. I hope we get along as the lecture progresses.
Today, we would be talking about the functional anatomy of the gastrointestinal tract. Digestion is defined as a process by which food is broken down into simple chemical substances that can be absorbed and used as nutrient by the body. Most of the substances in the diet cannot be utilized as such. These substances have to undergo a level of processing to get them ready for body to use. Now, let's go into the structural anatomy properly. Miss Guyton, you're up.
Thank you, Maester Teacher. Essentially, the GI tract is a hollow tube divided into major functional segments. The major structures along the tube includes mouth, pharynx, esophagus, stomach, duodenum, jejunum, ileum, colon, rectum, and anus. Together the duodenum, jejunum and ileum make up the small intestine, and the colon is referred as the large intestine. Generally, the organs of the GI tracts is divided into primary digestive organs and accessory digestive organs. Primary digestive organs. These are group of organs where actual digestion take place: mouth, pharynx, esophagus, stomach, duodenum, jejunum, ileum, colon, rectum, and anus. Accessory digestive organs. Helps the primary organs in the digestion process. They're included teeth, tongue, salivary glands, exocrine part of pancreas, liver and gallbladder. Associated with the tube or blind-ending glandular structures that are invaginations of the lining of the tubes, these glands empty their secretions into the gut lumen, example, Brunner's glands in the duodenum, which secrete copious amounts of HCO3. The major structures of the GI has many functions. The stomach and colon are important storage organs for proceed for chyme. The predominant function of the small intestine is digestion and absorption. The major specialization of this region of the GI tract is a large surface area over which the absorption can occur. The colon reabsorbs water and ion to ensure they are not eliminated from the body. Separation of regions of the GI tract is performed by a specialized structure called the sphincters. Blood supply to the GI tract is very important for them to carry out their respective function. However, venous drainage from the GI tract does not return directly to the heart, but first enters portal circulation leading to the liver. Wall of the GI tract. The major wall of the GI tract is formed by 4 layers: mucosa layer, submucosa layer, muscular layer, and serous layer. The mucosa layer, the innermost layer of the GI tract. It consists of the epithelium, the lamina propria and the muscularis muscosae. Epithelium. A single layer of epithelial cells line the mucosa. Within this cell layer are a number of specialized epithelial cells. The most abundant are cells called absorptive enterocytes, which express many proteins important for digestion and absorption of macronutrients. The cells are bound together to each other at their edges by tight junctions. The nature of the epithelium varies from one region to another depending on the function of that region. The surface of the epithelium is arranged into villi and crypts. Villi are finger-like projections that serve to increase the surface area of the mucosa. Crypts are invagination or fold of epithelium. The epithelium is mainly a stratified squamous epithelium. In the stomach and intestines, the mucosa is lined by simple columnar epithelium that facilitates secretions and absorption. At some places, the epithelium contains endocrine cells, enterochromaffin cells, that secrete local hormones. Lamina propria. Lamina propria is formed by connective tissues, which contains fibroblasts, macrophages, lymphocytes and eosinophils. This sublayer is rich in blood vessels, lymph nodes and lymphatic ducts, nerve fibers and capillaries. This layer supports the epithelium and also binds epithelium to the muscularis muscosa. Muscularis muscosa. Muscosa layer that consists of a thin layer of smooth muscle fibers. It is absent in the mouth and pharynx. Present from the esophagus onwards. The fiber is arranged in 2 layers. An inner and outer layers. Contraction of the muscularis muscosa in the stomach and intestines throws the musc's membrane into folds and ridges. In the intestines, the muscosal fold called plica increase the surface area for absorption. Contraction of the muscularis muscosa in small intestine produces local movements that facilitates the process of digestion and absorption. Submucosa. The next layer to muscosa is the submucosa. It consists mainly loose connective tissues and is highly vascular. Contains network of nerve fibers known as submucosal plexus or plexus of Meissner. Autonomic nerve fibers supplying muscles and blood vessels of GI tract closely interact with Meissner's plexus. In some region, most in intestine, submucosal contain lymphatic vessels. Muscular layer. This is called muscularis externa or muscularis propria. Consist of two smooth muscle layers. The inner circular and outer longitudinal layers. A nerve plexus is located between the muscle layers, called as myenteric or Auerbach. Auerbach's plexus contains fibers from the both divisions of ANS. Muscularis externa primarily control the motility of the GI tract that mixes and circulates the gastrointestinal contents and propels them along the lumen of the tract. Adventitia. Serosa. This is the outermost layer of the GIT. It consists of mainly a thin layer of connective tissues covered 6 with layer of squamous mesothelial cells. It provides external protection to the GIT. It's part of the mesentery that lines the surface of the abdominal wall and suspends the organs within the abdominal cavity. The mesenteric membranes secretes a thin viscous fluid that helps lubricant the abdominal organs so that movement of the organs can occur as the muscle layer contract and relax.
Wow, Ms. Guyton, you've done a nice job. Myself, would just talk little on the modification of the small intestine as our time is up. Villi. In small intestine, from the luminal surface, finger-like projections into the lumen. These are called villi. The surface of each villus is covered with an epithelial cell layer. Small projections arise from the surface of these epithelial cells, known as microvilli. Microvilli are collectively called as brush border. Folded muscosa, villi and microvilli increase the surface area of the intestinal luminal epithelial by 600-fold. Intestinal epithelial cells are continuously replaced by new cells. Center of each villus is occupied by lymphatic called lacteal, and capillaries, venules. Thank you for your